This is part 3 of our vaccine immunology series. In this chapter, we will be introduced to the immunological basis of vaccination, and the science behind some of the common vaccines in use today. Immunization is a process by which a person becomes protected against a disease, through vaccination. This term is often used interchangeably with vaccination or inoculation. A vaccine is a biological product that can be used to safely induce an immune response that confers protection against infection or disease on subsequent exposure to a pathogen. The initial response to a vaccine is similar to that of the primary response upon first exposure to a pathogen, slow and limited. Subsequent doses of the vaccine act to boost this response, resulting in the production of long-lived antibodies and memory cells, as it would naturally following subsequent infections. Vaccines vary in how they stimulate the immune system. Some provide a broader response than others. Vaccines influence the immune response through the nature of the antigens they contain. This includes number and characteristics of the antigens, or through the route of administration, such as orally, intramuscular or subcutaneous injection. The use of adjuvants in vaccines can also help to determine the type, duration and intensity of the primary response, and the characteristics of resulting antigen-specific memory. The immune response following immunization with a conventional protein antigen is depicted in this diagram here. First, when a vaccine is injected into the tissues, the protein antigen is taken up by dendritic cells, which are activated through pattern recognition receptors, and then traffic to the draining lymph node. Here, the presentation of peptides of the vaccine protein antigen by MHC molecules on the dendritic cell activates T cells through their T cell receptor. In combination with signaling through the B cell receptor, the T cells drive B cell development in the lymph node. Here, the T cell dependent B cell development results in maturation of the antibody response to increase antibody affinity and induce different antibody isotypes. The production of short-lived plasma cells, which actively secrete antibodies specific for the vaccine protein, produces a rapid rise in serum antibody levels over the next 2 weeks. Memory B cells are also produced, which mediate immune memory. Long-lived plasma cells that can continue to produce antibodies for decades travel to reside in bone marrow niches. Like in a natural infection, sometimes a vaccine dose is insufficient to generate enough antibody response to protect the individual. This is where the role of booster shots help. Booster doses of vaccine helps to stimulate the immune system to produce and sustain antibody levels above the protective threshold. This is clearly demonstrated in cases such as diphtheria, tetanus, pertussis and polio vaccines. The provision of 5 or 6 doses of these vaccines in childhood provides lifelong protection. Generally, there are 2 types of immunization, active and passive. In active immunization, a vaccine is given to prompt the body to generate its own response toward a particular type of pathogen. This kind of immunization produces long-lasting protection, but takes time to develop. In passive immunization, the protection toward a pathogen is transferred to the body through ready-made antibodies. The protection conferred by this type of vaccination is very rapid, but short-lived. Examples of this type of immunization include passive transfer of protective antibodies from mother to child through the placenta or breast milk, and in immunoglobulin infusions given for the treatment of certain disease such as rabies and pertussis. So what is in a vaccine? Vaccines contain biological products, or antigens that are either derived from an actual pathogen, or synthetically produced to resemble components of the pathogen. Some vaccines are live ones, and others are inactivated, or non-live. The difference between live and inactivated vaccines are listed out in the table here. In general, live vaccines contain weak versions of the virus, or pathogen that causes disease. Live vaccines provide a very robust immune response, and generate immune protection which are long-lasting. However, there is a risk of spread of the vaccine strain, causing disease in susceptible or immunocompromised individuals. Therefore, inactivated vaccines are safer, even though the immune protection it confers might be shorter and not as long-lasting. There are 3 main approaches to designing a vaccine. Their differences lie in whether they use a whole virus or bacterium, just the parts of the organism that triggers the immune system, or just the genetic material that provides the instructions for making specific proteins, and not the whole virus. Let us review the whole microbe approach. First up, the inactivated vaccine. The first way to make a vaccine is to take the disease-carrying virus or bacterium, or one very similar to it, and inactivate or kill it using chemicals, heat or radiation. This approach uses technology that's been proven to work in people. This is the way the flu and polio vaccines are made, and vaccines can be manufactured on a reasonable scale. However, it requires special laboratory facilities to grow the virus or bacterium safely, and can have a relatively long production time. It will also likely require 2 or 3 doses to be administered. Next, the life-attenuated vaccine. A life-attenuated vaccine uses a living but weakened version of the virus, or one that's very similar. The measles, mumps and rubella vaccine, the chickenpox and shingles vaccine, are examples of this type of vaccine. This approach uses similar technology to the inactivated vaccine, and can be manufactured at scale. However, vaccines like these may not be suitable for people with compromised immune systems. The third type of the whole microbe vaccine is the viral vector vaccine. This type of vaccine uses a safe virus to deliver specific subparts, or proteins, of the organism of interest, so that it can trigger an immune response without causing disease. To do this, the instructions for making particular parts of the pathogen of interest are inserted into a safe virus. The safe virus then serves as a platform, or vector, to deliver the protein into the body. The protein then triggers the immune response. The Ebola vaccine is a viral vector vaccine, and this type can be developed rapidly. Another way to make a vaccine is using the subunit approach. This method uses the very specific parts, or the subunits of a virus or bacterium that the immune system needs to recognize. It doesn't contain the whole microbe, or use a safe virus as a vector. The subunits may be proteins or sugars. Most of the vaccines on the childhood schedule are subunit vaccines, protecting people from diseases such as whooping cough, tetanus, diphtheria and meningococcal meningitis. Next, the genetic approach. Unlike vaccine approaches that use either a weakened or dead whole microbe, or parts of one, a nucleic acid vaccine just uses a section of genetic material that provides the instructions for specific proteins, not the whole microbe. DNA and RNA are the instructions our cells use to make proteins. In our cells, DNA is first turned into messenger RNA, which is then used as the blueprint to make specific proteins. A nucleic acid vaccine delivers a specific set of instructions to our cells, either as DNA or mRNA, for them to make the specific protein that we want our immune system to recognize and respond to. The nucleic acid approach is a new way of developing vaccines. Because of the COVID-19 pandemic, research in mRNA vaccines have progressed very rapidly, and resulted in accelerated development and approval for these type of vaccines. There have been many different types of vaccines developed over the years, ranging from those using whole organisms, to parts of an organism to small protein particles. Each of those have been proven to be effective and safe prior to approval for use in humans. Vaccine technology will definitely evolve to develop safer and even more advanced types of vaccines in the future. By completing our series of lectures on vaccine immunology, we hope that you would have understood important aspects of the immunological basis of immunization, and how vaccine technology harnesses the power of our immune system to protect our bodies from infections and diseases.